Hey everyone, welcome back to Grey's Gyan, your go-to channel for Grey's Anatomy Simplified. I'm really glad you're here. Today we're going to cover a very important topic, karyotypes, specifically, how human chromosomes are classified. This is high yield for exams and super important clinically. So let's get into it. So what exactly is karyotyping? Simply put, it's the process of characterizing the number and structure of chromosomes in an individual. Think of it as taking a photograph of all your chromosomes, arranging them neatly, and then checking if everything looks normal. Now, why is this important? Because many genetic conditions, like Down syndrome, CML, and others, are directly caused by abnormalities in chromosome number or structure. So karyotyping becomes a powerful diagnostic tool. The best time to look at chromosomes is during metaphase of cell division. That's when they're most condensed and visible. Prophase chromosomes can also be used for more detailed analysis. Point to the EMFISH karyotype image on the right. Now, how do we actually get chromosomes to study? There are four main sources. First, blood lymphocytes. This is the most common and easiest source, just a blood sample. Second, other body tissues. Essentially any dividing cell can work. Third, for unborn babies, we use amniocentesis, where we aspirate amniotic fluid from the uterus. This fluid contains fetal cells. Fourth, chorionic villus sampling, or CVS, a tiny piece of placental tissue. The big advantage? CVS can be done earlier in pregnancy than amniocentesis. Here's the step-by-step process, and this is a common exam question, so pay attention. Step 1. Cells are cultured and stimulated to divide. Step 2. Mitosis is arrested at metaphase using spindle inhibitors like colchicine. Step 3. Cells are placed in a hypotonic solution. They swell up, spreading the chromosomes apart. Step 4. Cells are fixed and mechanically ruptured onto a glass slide. Step 5. They're stained so we can see them clearly. Each step has a purpose. By the end, you have beautifully spread chromosomes ready for analysis. Now let's talk staining. This is where things get colorful, literally. The two most important ones are G-bands and Q-bands. Remember these for your exams. G-banding uses Giemsa stain after partial denaturation of chromatin. It gives alternating dark and light bands. Every chromosome has a unique G-band pattern, like a fingerprint. Q-banding uses quinacrine mustard, a fluorescent dye, and was actually the first banding technique developed. Then there's R-bands, the reverse of G-bands. C-bands highlight the centromeric heterochromatin. And T-bands specifically stain the ends of chromosomes, the telomeres. But clinically? G-banding is king. Let's look at real karyotypes now. On slide 6, you can see a G-banded karyotype. This is from a normal male, 46XY. Notice how the chromosomes are arranged in decreasing size, from pair 1 at the top left, all the way down to the tiny chromosomes 21, 22, and the sex chromosomes X and Y at the bottom right. On slide 7, we have a multiplex FISH karyotype, or MFISH, where each chromosome pair is painted a different color using fluorescent probes. This makes it incredibly easy to spot any mismatches or translocations. These two techniques are complementary. G-banding for routine use, MFISH when you need higher resolution. Now the classification. Human chromosomes are divided into 7 groups, A through G. Group A, chromosomes 1 to 3, the largest metacentrics. Group B, 4 and 5, large submetacentrics. Group C, 6 to 12 plus X, medium metacentrics, and this is the largest group. Group D, 13 to 15, medium acrocentrics with satellites. Remember, satellites contain ribosomal RNA genes. Group E, 16 to 18, shorter, with 16 being metacentric and 17, 18 submetacentric. Group F, 19 and 20, the shortest metacentrics. And Group G, 21, 22 plus Y, the short acrocentrics. 21 and 22 have satellites but Y does not. The key terms to know are metacentric, submetacentric, and acrocentric. They describe where the centromere sits. Now let's talk about the most powerful tool in modern cytogenetics: fluorescence in situ hybridization, or FISH. This technique uses fluorescent DNA probes that are specific to each chromosome. You can detect abnormalities so small they are completely invisible on routine G-banding. FISH is used to detect microdeletion syndromes, like DiGeorge syndrome, and specific chromosomal rearrangements like translocations. This is the most clinically important slide in this entire video, the Philadelphia chromosome. Here's what happens. There's a reciprocal translocation between chromosomes 9 and 22, written as T9, 22, Q34, Q11. Part of chromosome 9's long arm swaps with part of chromosome 22's long arm. The result? Chromosome 22 becomes characteristically short, and that's the Philadelphia chromosome, or PH1. This creates the BCR-ABL fusion gene, an abnormal kinase that drives uncontrolled cell division. Where do we see it? In over 95% of chronic myeloid leukemia cases, CML. And in about 25% of adult acute lymphoblastic leukemia, ALL. Here's the beautiful part. Because karyotyping identified this chromosomal change back in 1960, scientists could target it. That led to imatinib, also called Gleevec, one of the greatest success stories in cancer medicine. Okay, let's quickly recap the 6 key points from today. 1. Karyotyping analyses chromosome number and structure. 2. Metaphase is the best stage. 3. Chromosomes are in 7 groups A to G. 4. G-banding is the most widely used staining method. 5. FISH detects tiny abnormalities conventional banding misses. And 6. The Philadelphia chromosome T9, 22, is the hallmark of CML. That's it. If this video helped you, please give it a like. It genuinely helps the channel grow. Subscribe if you haven't already. And share this with a classmate who's also studying for their exams. Drop any questions in the comments below. I read every single one. I'll see you in the next video. Take care.